Hello, I'm Walter Schneider from the University of Pittsburgh, and I'm here to describe the 2009 Pittsburgh Brain Connectivity Competition. In the next 30 minutes, I will be giving an overview of the competition. Before we get started, I'd like to introduce you to some of the webcast features that will help you to look at the details of this presentation. You may need to turn off your pop-up blockers in order for the webcast to come through properly. Those on the Mac may need to add a plugin. If you wish to submit a question, which we hope you do, please click on the Ask button and type in your question, and we'll get to those at the end of the competition. If you would, please put the country from which you are asking your question and perhaps your name. In the download version, you'll be able to click on the slides button so you can move forward or backwards in the presentation, or you can speed up my talking. The button that's particularly important is the enlarge button in the lower right. If you click on that, you'll see the high resolution images of the presentation for the PowerPoint slides. We're going to cover the competition goals, then a short introduction to advanced fiber tracking, describe how the competition will work, what data you will submit, and then cover a variety of topics briefly, and then questions. So, brief overview of the competition goals. The goal is to advance human connectome mapping, that is, getting a full map of the brain areas and their connections. We want to provide focus, data, and educational materials to expand this research area. That we will have a quantitative competitive assessment of techniques. This will give you an ability to see what are the best techniques in the world for dealing with these problems. This will also provide visibility to the best groups in the world, and we will give out $18,000 in prizes at the Organization of Human Brain Mapping in San Francisco on June 22nd, 2009. Now, as a comment, we've done this competition twice before. In 2006, the Italian group of computational individuals who had never done brain imaging before took first place. In 2007, a UK group did well. I'm hoping this year the U.S. people place in the top level too. What is human connectome mapping? Well, Olaf Sporns has described the connectome as the connection matrix of the human brain, representing an indispensable foundation for basic and applied neurobiological research. However, he notes, the network of anatomical connections linking the neural elements of the human brain is still largely unknown. Well, what we want to do is work to better know the answer to what that is. It will involve identification of the functional brain areas, that is the location, surface area, volume, of tissue, and what are the connections both cortically and subcortically. It involves connectivity, both anatomical connection, that is the fiber connections, and functional, whether A influences B, and functional specialization of area, identifying the processing roles, the interpretation of subnets, interpretation of function. To illustrate this, it's a process of relating area, connectivity, and function. This is an example of the affect network that we have mapped with these types of techniques. So the first aspect is identifying the area. That is, for each of the areas within that network, identifying the volume and location of those areas. In the node and edge diagram, the size of the sphere indicates how much tissue is involved in these cortical areas. It also means identifying the connections. That is, the cross-sectional volume of the connections between areas within that set. We can then get a measure of the amount of fiber pathway of the track between areas, which can vary dramatically. And then we can look at functional connectivity, things such as resting state activity or Granger causality within the network of areas we've defined anatomically. So in this competition, we are going to use MRI-based methods of connectome mapping. That is, we will be getting structural data, diffusion data of the fiber tracks, and functional data of the brain activity. Note there are many techniques that provide useful information, from animal work, human multimodality imaging, TMS, lesion studies, dissection, etc. These, however, will not be the focus of this competition. Let me provide a short introduction to advanced fiber tracking. There are two key challenges that must be solved in order to make a connectome map based on human MRI data. The first is we need to follow fibers through multiple crossings, and the second is we need to identify the anatomical and functional boundaries of the cortical areas. So let me give you an analogy. If you were trying to understand a modern computer system like your laptop, one of the things you might want to do is create a circuit diagram of all the connections. However, current boards have like 14 layers, and if every time you try to look at the connections, if one layer crossed underneath another, it would be of limited utility. The graph on the left is an example of an 8-layer board, and the figure on the right shows that if I try to follow the tracings, I basically see that they leave the chips and then they're lost. This is the problem of fiber tracing through crossings, which has been a major challenge for diffusion-weighted imaging. Diffusion-weighted imaging uses MRI to label molecules and then tracks their diffusion. If they are diffusing in all directions, isotropic diffusion, as happens in intercellular fluid, you can't identify the direction. If they are in a confined space, such as tubes, you get an isotropic diffusion, that is, it is elliptical, and you can then use that to identify the direction of the fibers. This technique is done by taking a number of directions, the minimum is 6 directions, to identify the diffusion directions that the fiber might be taking. However, there's a serious fiber crossing problem. On the left here is an image using diffusion tensors with a small number of directions, and you'll see that in many of the places you have these spheres because two fibers crossing look in this kind of method with small number of directions, just like an isotropic diffusion, and hence you can't tell the direction. In the last 2 years, there have been dramatic progress in our ability to look at diffusion through these types of crossings. And in this competition, we will be providing data that supports this kind of analysis, where if you now look at, for example, the upper row, you can see a combination of fiber crossings. This illustrates an example of this from various images, where the diffusion technique can now resolve double and triple crossings of the fiber path. Given that, we can then make a map. Here's a slice that we collected, and we can zoom in on a given area within that process. We can identify the fiber tracks, and if I zoom in further within that set, and get the individual directions of multiple tracks within that system by getting these high-resolution mappings of the fiber tracks. Now, we can do that through multiple sections of the brain and build fiber tracks out of that result. I'm going to switch here to an illustration of this process. What we have here is a mapping of 250,000 fiber tracks that go through the central plane within this individual, which is part of the competition data set. And I note those of you who have looked at diffusion tensor imaging, note this is not the extracted corpus callosum that you often see. We see fibers going all the way from source to destination throughout the brain within that process.